So is this one on?
Yes, they should be able to hear you. Everything should be ready, so you can get started.
Yeah. So hi everyone. So my name is Victor, and thank you for this opportunity. I think this is the last SMAX meeting.
We're gonna have second last.
Oh, second last.
Yes.
Okay. Oh, I thought I'm the last one. So basically, in today's talk, what I want to do is just kind of take you through a review of what we have done regarding the study about the raw GTPS models, and then I'll give you also tell you about the current focus and some of the future opportunities about this work. And so to start us off is just to kind of motivation. So we are studying within this area of cell migration. And so we know that cell migration plays a vital role in many processes within the cell, ranging from tissue formation, wound healing, organ development, and then we have this about the cancers. So here I show you a picture of, it's just a snapshot from a video of where neutrophil is chasing a bacteria. And so you have a bacteria, you have a red blood cells, and then you have a neutrophil. So when the cell is migrating, there are many things involved. So this is a multi-step process involving the protrusion of the cell there, the front, and the destruction of the back. So we need to understand why the cell migrates. And then understanding this, we have to look at different processes. One way is to look at the signal transmissions within the cell, which are controlled by what is called the raw GTPases. So these molecules reorganize the cell cytoskeleton such that the cell is able to maneuver and do different or various processes. So can you confirm if that one is moving? Because I just it's stuck.
Oh.
It is not.
Yeah,
okay.
can screen share on your laptop.
Okay. Yeah.
Right, it's a screen share.
Okay.
So I didn't even know you could pause the screen share. I don't know.
So how do I?
Wait, just unshare and share it again.
So Zoom.
Oh, but now it's green again. But it's not. Can you just scroll down just to see if that moves? If that moves, if you scroll down, okay.
So did I?
Let me making it full screen again. And then it pauses.
That one does not go to the full screen. So can I stop share? Okay.
Maybe try sharing. Yeah, with the after turning on the full screen.
So how do I go back to Zoom if I have full screen?
Just I think you can go down. Yeah. Tab. Go down. This appears.
Okay.
Yeah. I think she just said this full, the full. Okay.
Okay. Yeah. Thank.
No, there's a. Okay.
Now it's not.
Just once click on the slides with your mouse.
Okay.
Just make sure you're there. And I'll try to see if they can.
Okay. Okay. So I just repeat. So I was showing here a snapshot of a migrating cell, which is a neutrophil chasing a bacteria. And I said there are different processes involved in this part. When we want to understand this, we can look at the signal transmission within the cell. And one of the proteins involved are called the raw GTPase proteins. So these proteins control the cell cytoskeleton by reorganizing it such that the cell is able to maneuver and perform different processes. So there are different types of raw GTPases, but the most studied one is called the CDC42, the RAT, and then the RAW. So this CDC42 controls the direction of migration. This controls cell protrusion at the front, and then RAW controls the contractility of the cell. So our focus here, we are going to focus on this molecule called the Rho, which controls the cell retraction or cell contractility. But before going to look at this, so these molecules act as on and off switches, where they transmit signals when they are in active form, and otherwise they can be turned off and then they are in active form, inactive. And so we see that they are here. We have an active Rho or GTPS. We have an inactive Rho controlled by the gap and then the gap. So this is just the definitions. So this is just a brief introduction about the cell migration and then the Rho GTPS that we want to look at. So my focus today is just to give you an overview of what we have looked at so far. So I'm looking at a signal pathway composed of Rho, GEF, and myosin. And so in this work, there are a number of work which are already out, others are on the way, and others also in preparation. So the work started with the group in Max Planck Institute in Dortmund and University of Duisburg, where they started this experimental setup of this network. And then followed by, we developed some models to describe this behavior. Models were refined in this, and then experiments done, and then parameter fitting was done here. So from here, we are now trying to understand these systems, either as a mathematical model and also as a way of trying to explain this biological behavior studied in these experiments. So one of the work is the work by Kusanagi, which was accepted in Royal Society Open Science Journal. And then we have also another one by Nanda, which is currently in review in mathematical biosciences. And so here, the difference is Nanda alters some of the mechanisms we study in this network and then try to see if we can explain the same behavior in this model. And so currently, we're also preparing another one here. We are now digging deeper into some of the regions where you have biostability and then we have what was electrodynamics. So I'm just going to give you a brief review of this. So starting with this work of Greishal. So Greishal were the first to look at this network composed of Rho, myosin, and the GEF. And they identify this self-organized system composed of Rho and the GEF through the positive feedback, and then Rho activates this actomyosin or myosin, which in turn inhibits the Rho. So what they identified here is there are two types of myosin, myosin 2A and myosin 9B. So myosin 2A inhibits Rho by inhibiting the GEF H1, and myosin 9B acts as a gap for Rho, so just inhibits Rho directly. So they show that the interaction or coupling of this network and matrix elasticity can explain this propagating process of cell contraction. So the focus here was to develop kind of models to describe this behavior. So the Rho myosin interaction. So based on this, we now followed up with, which is the work in my thesis, by developing a series of mathematical models to describe these dynamics. So if you look at the previous network, we have three proteins or three components. We have the GEF, we have the Rho, and then we have myosin. So both of these exist in either active form or inactive form. So we can form 6 species mathematical models. But because of these molecules are conserved, we can reduce this into 3 different equations or ODEs. So we can have different mechanisms or different mathematical assumptions or biological assumptions and coming up with different mathematical models. So we derived different mathematical models, but in the thesis, we only have 3 models, which we labeled as GRM1, GRM2, and GRM3. And the focus of this was just to try to understand what are these temporal interactions of these proteins and then understand their behavior. So this was just the structure of what we went through. Develop models from experiments, we formulate the models based on the experiment, try to analyze, perform some analysis, pilot estimation, then we go back. So then we try to formulate these models, try to refine them. We just had another extension in the test just as a proof of concept for the spatiotemporal dynamics. So based on this, the experimentalists were not happy about the bistability, that they were not able to observe bistability. And so the first two models we formulated, they're not happy about them. And so that one gave rise to this study about in Kamps et al., where now they tried to dig deeper and try to understand the biomechanical interactions of these proteins. So try to get the assumptions. And so in this, one of the models which is presented here is also just presented in this work, but in the general format where we include all the processes, including the background activities. So also in the experiment, since they were not able to measure the background activities of either the raw or the myosin, those were neglected. And that model was compared to analysis and then experiments, and then these are the results. So this is just a graphical abstract on the article, on this. So after this work, their work here also involves the spatiotemporal dynamics, but they are using the cellular automaton. So we wanted a model of where we can perform mathematical analysis. So we have mathematical analysis. And so in that case, we had to develop some partial differential equations. So this is just an outline of what, again, just rewriting the same things. We are having three proteins, GEF, rho, and then we have myosin. And if you check in this, I only have one pathway from myosin to the GEF, and then we have neglected the pathway from myosin to the rho, which is a gap. Because in the experiments, in the Kamps work, they say that myosin 9B has very low expression levels compared to myosin 2A. So they didn't test that one, so it was just assumed and then ignored. And this is the work which has now been done by Kusanagi. We know we have a 6-species mathematical model, which might not be easy to perform mathematical analysis. And so to try to understand some of the behavior which can exhibit in this model, we try to first just narrow down to a 2-species model, and then see what we can learn from these 2 species, and then we can rescale up. And so what's happening is we have a 6-species mathematical model. Because of conservation, we can only have the active species, so the 3 equations. And now by applying quasi-steady state approximation, we can neglect either the GEF or the rho. So in this work here, we assume GEF comes as just as a function quasi-steady state, and then you have the 2 species R and M, which we can write as what? As U and V. So if you forget about the biology, we are just having 2 species interacting, U and V, and then we can have all these assumptions where we can write now the reactions involved in this part, and then we study. So this is the work which was done in Kusanagi's, which is in Kusanagi's paper, just reading the simulations in a different way, but they are just the same results which are in Kusanagi's work. And we identify 5 different dynamic regimes. So when you vary parameters, we can have either a unique steady state, which is stable, or we have this other unique steady state also, which is stable, but we will see that this one can be affected by the diffusion. The first one can't be affected by the diffusion. Here we have a unique steady state that's unstable, and so we have oscillatory dynamics or limit cycle. On these other parts, the gray and the yellow one, we are having coexistence of different states. On the gray, we have coexistence of 2 asymptotically stable steady states. And on the yellow one, we have coexistence between a stable limit cycle, locally stable, and a steady state. So this is what was presented in Kusanagi's work. So you see that the current focus now, we are focusing on these 3 regimes. We have mathematical analysis for this. We're having the Turing-driven instability about this. So following Kusanagi's work, we also now have Nandua's work. So the main idea about Nandua's work is that Nandua now brings in this pathway from, I think this B is myosin, from B into A. So in Kusanais and CAMPS, they neglect this myosin 9B. But so in Nanda wanted to see what is the effect of bringing in this other inhibitor as myosin 9B. And also, Nanda has also different models. But in these models, he tries also to simplify some of the mechanisms we have in the previous models. Where you have the heat function, just tries to model the mass action. And so these are the results. This is just an abstract. We have a mathematical model. We also have similar dynamics where we can have bistability or you have a unique steady state. Also, we can also have oscillator dynamics. And so we have these are just the Turing spaces when you have the Turing-driven instability. We then perform simulations both in 1D and in 2D. And these are some of the results you can have. So Nandwa isolates modes and then tries to associate these solutions to different modes which are isolated. Yeah.
Sorry, is this still a two-variable system?
Yeah, it's still a two-variable system. Yeah. So if you notice that I try to avoid writing the exact interactions, right? I'm not just labeling them as F and U. But so if you check the Nanda and Kusanai, they are different here, but still a two-variable system.
Two variables in Nanda.
Nanda is still the R and M, where the graph is taken to be equal steady state. Yeah.
Those 2D simulations are like on the forums?
Sorry?
Are they like periodic over here?
Boundaries.
No, we just have the zero flux, Newman, yeah, zero flux. And so following this, so this is just a summary. So we formulated different mathematical models. What are some of the properties? We see that all these models can give you all these properties. But in some of them, you cannot have this and certain bistable existing in the same parameter regime. So you have either transition from a stable to oscillatory back to stable, or you have transition from stable to bistable and then back to stable. But in one of the models which I have in the thesis, we show that you can have this transition across where you have all these regimes. You can sweep through the stable, oscillatory. We have excitable also, which is just still the stable, and then back to bistable, as you can see in these bifurcation diagrams. So this is what we have done. As I said, now the current focus is trying to dig deeper into these bistable regimes and then oscillatory regimes. And so I just summarize here now from Kusanai's work. So these are the three regimes I'm digging in. We have bistability, we have oscillatory dynamics, and then here we have existence of oscillations and then stable steady states. So I'm going to focus more on this part where we need to understand what is the effect of diffusion into these stable limit cycles. And to study that, I look at this Floke theory. So assume we have a system, a reaction-diffusion equation, having periodic solutions which are now spatially homogeneous. And so if you have these are periodic and spatially homogeneous, they must satisfy this type of OD, where now these are periodic with the period, which I call here T. So the aim is first to understand how do we test the stability of this limit cycle. And so we can linearize this system about the steady state. And then we get this. I just skipped a lot of steps. But so when you linearize, you get this part where J here is the Jacobian evaluated on a limit cycle. And so we know that here J is not constant anymore. So J is just time dependent. And if these are periodic, J will also be periodic with the same period T. So by Floke theory, notice that this system, J is periodic, but we might have that the solution is not periodic anymore. We might have periodic or non-periodic solutions. And so to test this stability, we look for what we call these fundamental matrices. And then somebody can show that if you have this system 1, if xt is a fundamental matrix, then xt plus t is also a fundamental matrix. And this one can be proved. And so this relationship, the connection between xt and xt, they are just like a scalar multiple of the other one. Xt plus t is equal to xt plus multiplied by an unsingular matrix C. So that one allows us to define this matrix M, which we call the monodromy matrix. And so this matrix just represented by what? The inverse of the first fundamental matrix, and then multiplied by this after one period. So this M depends on the choice of fundamental matrix you have. However, what we notice is that these eigenvalues of M are not dependent on this choice of this. They are a property of the Jacobian matrix. So they are just constant always, whichever fundamental matrix you take. So these eigenvalues here, which we call the Floquet multipliers, are the ones which are used to analyze the stability of these periodic solutions. And so we see that since we are on a limit cycle, one of the Floquet multipliers will be 1. I think it's just saying that if you have the transverse perturbation, then they are not affected in any way. So for the limit cycle to be stable, we expect that now the second one has to be less than 1. If it's more than 1, then the limit cycle is unstable. So we need to carry this forward and assume in this case that the limit cycle is stable in absence of diffusion. That means if you don't have diffusion, then you have that this rho 2 will always be less than 1. Yeah.
In the monodromy matrix,
Yeah.
little t's usually taken as zero.
It's just the initial time, so it can be 0. So it's just the initial time of the solutions.
Right. Okay.
Yeah. As long as wherever you start from, as long as you just measure the x at time t and then x at time t plus t, which for simplicity we just take t as 0. Yeah. Yeah.
Because I guess otherwise those Floquet multipliers would depend on t if you want.
But so what I saw is wherever you start from, even if you pick anything in the middle, and as long as you take xt at the point t and then xt at t plus t, you will get a different matrix, but these are constant irrespective of m you get.
Oh.
Yeah, yeah. So this Floquet does not depend on the choice of this. I think there's a theory we can check. We see that the determinant of this m is exponential of the trace of the Jacobian matrix, the integral of the trace. So it's just something which depends on the trace. And the product of this gives you the determinant. So they're just depending on the trace of exponential of the trace of Jacobian. So now having seen, or assuming that the limit cycle is now stable in absence of diffusion, we now extend and see, or try to study what's the effect of adding diffusion to this limit cycle. And so by adding, we follow through the same process. We linearize now the full system around the limit cycle. And then we get this, where J, T is the same Jacobian matrix we have on the ODE system. And D is now the diffusion matrix, if we non-dimensional. So we have 1D. And so we can apply separation of variables or eigenfunction expansion and show that this system can be written in terms of this where k are the wave numbers. So if you set k is equal to 0, then you just go back to your ODE system. So the task here is now to check that for each set of parameters, can we get a k or k such that rho of k squared is now less than 1? If yes, no. We need to check to be sure that at least there exists a k where this becomes more than 1. Then in that case, it means now the limit cycle has been destabilized. If there is no such a k, then we are not destabilizing the limit cycle. So it remains stable in process of diffusion. So we applied this first to just selected parameters from the two regimes we identified as, where we have a unique steady state that is stable, unstable, and then you have resolution dynamics. And then the second case where you have existence of oscillations and then a stable limit cycle. So if we apply and then we are doing this computationally. So we try to make sure that we go to a limit cycle computationally. First check that this limit cycle has to be stable in absence of diffusion. And so we can see that when we check, we see these are the Floquet multipliers. One of them is 1 and then there's 0.05 976, which is less than 1, it shows that this limit cycle is now stable in absence of diffusion. And now try to vary diffusion coefficients and check to see that if there exists at least one wave number such that this the flow multipliers become more than 1. And so if you have the simulation here plotting maximum of the flow multipliers against the diffusion coefficients, then you see that when the transition here somewhere around 33.22, then all of them become more than 1. So it means for diffusion coefficients above 33, we can destabilize this limit cycle. Here just to the same, but just to show this part. And this is now bring in the gamma. So the role of gamma here is just it translates.
What are you plotting in the middle one? So
this?
No, I understand that, but the second one. So you are isolating the
yes.
numbers.
Yeah.
That's important.
Yeah. So here I'm isolating the wave numbers to see which ones are destabilized. Yeah.
And little d is the ratio of diffusion of what to what?
So the ratio of diffusion of myosin to rho. Yeah. Or in this case is V to U, because I'm not doing general variables U and V. Yeah. And so we see that the modes you isolate change when you vary gamma. So the role of gamma is just to, it kind of changes the modes you are isolating, but does not change the stability. So it keeps the same part, but just sweeps it across. And so if you check these results, they are just similar to the Turing analysis, the Turing-driven instability, where now we are talking about the eigenvalues more than 1. But so you're just talking about the flow multipliers. So I also did this on now where we have coexistence between the 3 steady states, and stable one, a saddle, and the stable one. And just when you do the same analysis, you also get this, that you have is stable in absence of diffusion, but becomes stable for some diffusion coefficients. And so the task was now to generalize this to where we are now varying parameters and then check the regions where we can exhibit these dynamics. And so when we do this, I presented preliminary results, because these results, they need to be refined so much. So for me to get this one, my laptop, I think, was running for 4 weeks, non-stop. So what happened now, I'm now using Antida's cluster, the VM machine. So I submitted one on Monday, but I think now it's at loop 117 out of 500, but on Monday. But I think it's a bit faster than my laptop.
Can you parallelize it?
That's what I did. And so it's doing on, because I'm doing the parallel on the J loop. So it's doing 150 simulations at a go in the J loop. Yeah. Maybe I can do double parallelization on the J and I, but I've not done that one. I'm only doing on the J one. Yeah. So what is the idea we get in this? We have that the orange here shows that the limit cycle is not destabilized. So I just put a legend. And then NLC means here now we don't have a limit cycle. So we either have a unique steady state or we have a bistability coexistence between the two stable steady state. Here I define period ND because I was having issues on the convergence when you set large time steps for each one on the laptop. And so some regions you find when you have this purple color is that you cannot get the limit cycle. And then DDLC is now where we are destabilizing the limit cycle. And so we see that as you transition now from the D values, D1, D10, you see that the region of where the limit cycle gets destabilized starts increasing. So this is the part, yeah, sorry, yeah.
The 2 parameters on the...
Oh, so I'm just taking the 2 parameters, A1 and A5. I tried to...
I don't remember what they are. So roughly, what are they?
Kusanai, can you? But they are just basically the same parameters in Kusanai's work.
Okay.
I think A1 is related to the GIF concentration, right?
Yeah,
the GT,
yeah.
the G total, yeah.
Yeah.
And then one of these is an activation of myosin. A5 is activation of myosin, yeah. But we could, studying this mathematical problem, we could do this on any of the parameter combinations, bias parameter combinations. So these are the results we obtain. So we have to validate this numerically. So I have not validated for the new simulations because I still have to wait for them to finish. So otherwise, what we had, I will first of all just review what we had with Kusanagi, showing that for this, if the steady state is not unique, nothing happens when you add space. That's the first type of steady state. And in the second one of this type of the steady state, which exhibits or admits the Turing-driven instability, we see that we can start from a uniform steady state, but in the process of diffusion, we can destabilize this and then we end up getting these partial patterns. Just here we are having an L2 norm. So in the third case, this is now where we have a limit cycle. So in limit cycle, we can have different behaviors. We need to understand them into details. But one way is we can still maintain the limit cycle, but it changes positions depending on the spatial location, which is still shown in this part. We're just having the same limit cycle, but the limit cycle just changes the behavior. But on the other end, I think I didn't include it. One end is the limit cycle just gets completely removed, and then we end up with the spatial patterns. So I'll see. I don't know if this can be linked to the gamma when you are reverting the gamma, and then maybe when you have that gamma for some gamma values, you don't isolate any integer modes. I don't know if it has a connection. That one I have to check. Next, we go to this other region, which we call region 4, the coexistence between the two steady states, stable one and unstable one, also this stable. We can show that this stable steady state is of type of the first one. Yeah.
Sorry for digressing, but what was the region that we were working on the previous slide?
Oh, so I call them region 3, where we have a unique and stable steady state. So you have existence of limit cycle.
A unique stable steady state.
Unique and stable.
Okay.
Yeah. Region 1 is unique, stable. Region 2 is also unique, stable, but it admits the Turing-driven instability. Region 3 is unique and stable, so we have a limit cycle. Region 4 is coexistence between the two stable steady states. We can show that this is of type the first one, so it does not affect. It's not affected by diffusion. So diffusion only affects this other steady state, of which if we introduce diffusion, you either migrate to the upper one or you end up forming the spatial patterns. And so here, you see here we have a spatial pattern. So when this is destabilized, and on this other second graph, we see that we destabilize this, but now we end up migrating to the above steady state. Here. We do the same one on.
But so it's independent of what you choose your parameter, your initial conditions, because it's important.
So here I kept initial conditions the same and only changed the gamma. So that's what I'm trying to understand. Why does gamma affect the switching to this steady state or bring about the spatial patterns? The only parameter I'm just varying here is only the gamma, and then
so you choose them closer to the steady state inside the limit cycle?
Yes, I choose them inside the limit cycle. This steady, so I choose them within the region of attraction of this, just very close to this steady state.
And how is the transition? Is it a smooth transition, or is it do you have jump conditions across the transversal, I guess, because there should be some region of attraction for each of these steady states?
Yeah. If it's a jump, I don't know if that one we can tell it from this diagram, where we just start from a small perturbation around the state.
Trajectory of the loci of how the system imagine from how is not. You would expect it should go to the uniform steady state.
Yes.
Close to,
yeah.
But it's going past the limit cycle, so it's going, through that region where it's supposed to be attractive, but it's going to the completely different uniform steady state.
Yeah.
And so the question is how can we not plot this transition? I would like to see how it is.
Okay.
because is it smooth? Is it a sudden transition? I don't know. Is what kind of, if you have the hysteresis, some type of hysteresis, then you have maybe jumps. I think it's how does it change from one state to the other? Is what I want to understand.
Okay. Maybe I can try to look into this.
Yeah. I would be surprised if it is very small. Probably, I don't know.
Yeah.
Can you follow the initial conditions somehow?
Okay. Yeah. I can look into that one. And so this is just the same one. And now we have the same behavior for the region 5, but so region 5 we are having conditions between a limit cycle and a steady state. And so here when I do all these simulations, I make sure that I start them within the limit cycle. And so in the process of diffusion, you are kind of crossing the limit cycle and then you go back to the other steady state. But I think because we say that the steady state, the diffusion now makes it unstable, so it means we cannot go to that limit cycle anymore. But just also in the previous results, when you destabilize this, you can either end up getting the spatial patterns, or you also have the transition into the above steady state. So this is what we have so far. I will try to refine this based on the new simulations when they are completed. But so following this work, what else do we need to do? One way is we are now trying to scale upwards, trying to go back to the 6 species. And so this is now, in the previous models, I now give you here the full equations. We assumed a quasi-steady state on the first species. This is this second one, which is the GEF. And so we have been dealing with the first equation here and the last equation. So if we disregard and then we don't apply the quasi-steady state, we now have the 3 species, which already Kwasana is looking at this. And I'm thinking that now we can apply the theory by Anutida for the 3 species and then try to see if we can generate the region, so try to identify the Ds of instability for this one. I think it's a paper which was submitted already. So this is already in progress, and the advantage we have here is that this system, when you set this B3 to be 1, then has the same status as the previous one we have been studying. So they just have similar steady states, but different characteristics. And also, I think Kwasana has some preliminary results. You can show that their bifurcation diagrams are qualitatively similar for some parameter regimes. Another extension we look at, so this is just based on the discussion with the experimentalist, is we are having a 6 species model. We are trying to simplify so that we can get a minimal model that can still replicate the experimental observations. If you check the 2 previous systems you have been looking at, we replicate the dynamics qualitatively, but we are having an issue with the D. So D is not what is experimentally feasible, because myosin diffusion is much slower than the rho. So we think that by scaling upwards, we can still get the same dynamics, but now with experimentally observable or acceptable diffusion coefficients. So what we have here, they assume is that the GEF and rho, they're kind of similar in their dynamics. And so we can disregard one of them, just disregard either rho completely or the GEF completely. And then we now end up with a 4 species mathematical model with the diffusion now, which we can study. But again, this can be simplified further if you want to simplify still to see if we can still get the same one, but now with the diffusion. And so the assumptions where the experimenters are mentioning, or the justification for simplification might be this, that concentration of myosin is very large compared to the rho and GEF. And so this one they think might not maybe affect this dynamics. So can we assume a constant or homogeneous concentration of the inactive myosin so that we keep the 3 equations G, I, and then M, but this one just comes as a constant or a function. The other one is that GEF H1 active diffuses much faster than the rest of these. So we can assume that GEF is in what? Is in quasi-steady state. So we also still end up with 3 different equations, but keeping inactive molecule. So they believe that for us to replicate the experimental observations, we should have at least one of these inactive molecules just playing as also another inhibitor, as opposed to where we have previously, we are just looking at 2 molecules, but which all of them are active molecules. So trying to simplify, but maintaining at least one of the inhibitors. The other extension, which we also look at, is when we did this, they assumed that since myosin activities are much slower, so they ignore this myosin defection, that's the myosin. But so they believe that there are some experimental observations where this assumption can't be ignored, and so we can try to check or bring in this effect of this protein carop and see how that one affects the myosin. And I did the same one. There's a way they explain that it affects the signal, which again also affects what? Affects the rho. So we are just modifying the equation we have by introducing some constant signals in the rho, and then introducing constant signals in what? In the myosin. And there is an approach here which we have to look into that started in this other paper, which applied to a similar model. And so if we have this model, can we still be able to replicate the experimental observations here?
What is that little V over there?
I think it's something which has to do with the velocity. So myosin is also, I think the defection of myosin about this part. Yeah, but I still have to check exactly to see the implication of this part and how we can use it. So these are some of the extensions we have. Let's say if this works, and if the assumptions we have here do not change or replicate the experiments, can the same assumptions now, when applied to this model, give us some experimental observations? So these are some of the work that we need to do. And I think this is what I had for you today. Yeah. Just to point out, in this picture, this is where everything started. This work, I think, started around 2014. In the first paper, experiments were trying to publish in the whole of this results, theoretical and model, by just taking the Tyson's model directly and then into their work. But I think they were not able to. That was the main critic. And that's when now they brought in Anotida and Stefan to try to look into the modeling part of this work. And so when I started my PhD, now that's when I got into this work. So the experimenters are here. We have a live document from Max Planck Institute, and then you have Perihan Nalbert from University of Duisburg. Those are in charge of the experimental observations. This was their student during the PhD called Rupuja, also involved in the experiments. The others you see, like the camps, the Greisel they were not part of this, so they are not in this picture. But that's what they're involved in this one. Okay, yeah. Yeah.
When you were doing the Floquet cycle analysis, did you ever get, like, all the stuff you showed, you had your second eigenvalue crossing at 1. Did you ever have period doubling where it crosses at -1?
Maybe I need to check, because so here I'm just studying the Floquet theory, just try to identify the Floquet theory, and then see where they are more than 1. I can maybe try to check, as you mentioned. So when you do simulations, try to check at the point where now both of them are 1, 1, right? I think if that's what you mean.
Yeah, but you can also have -1.
-1.
You can have one negative.
Oh yes, yeah.
And when one of them's negative,
Yeah.
like when you're largest, ignoring the translational.
Okay, yeah.
If there's a -1, you get a bifurcation.
Yeah. So maybe what I can do, because here I'm just putting the maximum, I can try to plot both of them, row 1 and then row 2, and then try to track them and then see whether they are 1 or -1. Yeah.
It might not ever happen.
Yes, yeah. Yeah, since I have them, so I think it's just a matter of plotting them and then try to check. I'll try to look at that.
There are a couple messages on the chat, on the Zoom chat, if you want to look. Some of them are just saying good job, but.
Okay, maybe you can check if there's
We'll check it.
if there's a question. Yeah.
Or if they have, they can also unmute themselves.
Yeah. Then there's.
Don't be shy.
So this
Yeah.
Why do we have to worry about the cytosol and the cortex if it is not taken into account?
Oh.
Because the inactive and the active, they generally are resident in
Yes.
different domains.
Yeah.
So does this matter at all?
That one, honestly, that one I'm not sure if it can have an implication on this part. But maybe it might matter because they reside in different regimes, and then they also. I think maybe that one you are in better because you have dealt with the bulk surface. So maybe you can see the implication of either having the same domain or having the coupling between the bulk and the surface. Yeah. Yeah.
I think it will be one of the to-do projects because I think it will be really nice to look at the compartmentalization of these molecules.
Yeah. Okay.
Not in the same way as you think of bulk surface.
Okay.
But in terms of the cortex cytosol domains, there are still planary domains, but you just have these two different domains where you're coupling boundary conditions on the interface between the cortex and the cytosol.
I think something similar to what Guzman is looking at, right? Yeah, I think might be similar. You're having, yeah.
I don't think she's just looking at the cortex. Yeah.
Oh, okay. 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 Yeah. Then I was thinking, I don't know, because there was, if you check at the work of, when they mention here that they are able to replicate this, is it something, so when we have this, and then you can bring the matrix elasticity maybe by something of kind of mechanical forces,
yeah.
Is it something that can be done with, like the solution Jupyter does?
But do you have a continuum mechanics model?
I mean, it supports balance model. It's continuous.
Yeah, you try to replicate this using the force balance.
But you're talking, okay, what is the metric? Are you talking about the extracellular metric?
Yeah, because I think they have this matrix elasticity, extracellular matrix, the interaction.
So you're talking of how the cell is interacting with an environment that obeys some continuum mechanics model.
Yeah.
Which can be solved still in the model. Because those walls that I had, they're technically being solved. It's just that the walls are moving.
Sure. Then you will need a continuum model for the extracellular metric.
Yeah.
I'm assuming that I think this can be done. But
because what they mentioned
that different. Yeah. Sorry.
Okay.
Yeah, it's a different from.
I think it's different because what they mention is that if you couple these two, then you can reflect these activities of rho onto the cell and how the cell is contracting and then retracting.
Absolutely.
Yeah.
But and how the mechanics feed into the biochemistry, exactly as you are. Yeah.
Maybe this is what we can try to ask them to see if they can give us the feeding here.
Yeah. I think it will be also a nice project to look at. Definitely.
Yeah.
But then in that case, you really need. It's not so clear to me that this could be just done for stationary cells, because at the moment everything is stationary, and also I'm not so sure if this is something you might want to ask them.
Yeah.
When they do this continuum, are they looking at a stationary? How does adhesion? I mean, I'm thinking of how the cell is adhering to the substrate.
Okay.
Is it migrating? Is it just stationary, or they put it on and then what are they measuring? Is it the contractility, the metric contracting, or what is it?
Okay.
I'm not sure.
Okay.
It's not so clear to me.
Okay.
I would look at this as a migrating cell, how it's migrating, and how the extracellular metric might hinder or promotes its migration.
Okay. Yeah.
But again, I think it's a different framework.
Yeah.
Yeah, I like this part, but I think what you didn't show is you have the limit cycle.
Okay.
I think you need it. Wait, go back again. In one of the slides,
This?
Yeah, no, the other one. Yeah,
Yeah, okay.
this ones, I think this is quite important. The last two,
Okay.
you really don't describe, but I think that's really very important because the blue one is the ODE system. So
Yes,
this is the solution of the ODE.
Yeah.
That's the limit cycle of your ODE system.
Yeah.
And if you take now your initial conditions, which this is what I was trying to also intimate. So your initial conditions are very close.
Yeah.
And that's the pink one. But you
Yes,
cannot
yeah.
see because it's almost embedded in the blue one, but there's a trajectory of, it's not a limit cycle yet for the PD. So the pink one is the PD, is the solution to the PD.
Oh, yeah.
And if you follow it, initially there's no limit cycle. It only forms much later.
Yeah.
It's like, if you look at D, for example,
in this region.
exactly.
Yeah.
There is no limit cycle.
No limit cycle.
It's a formation of a pattern. It's actually the system is trying to form a pattern. That's the first thing.
Yeah.
Is the pattern formation. It recognizes that the diffusion-driven instability conditions are satisfied, and it wants to go to this pattern. So that's the first thing. The formation of a pattern.
Yeah.
Maybe I asked you to plot the L2, but it's there. But I asked you to enlarge this.
I did not because I was waiting for this other one.
Yeah. Fair enough.
Yeah.
But if you look in F, you can point to the first delta.
Here.
You see the peak.
Yeah, the small peak here.
The L2 norm, that's if you were to zoom in, the L2 norm is going up and down. And that's a reflection of the formation of the pattern. So the pattern is formed, that's the first verb. The second one is, it becomes not completely stable, but there is another transition of the pattern becomes oscillatory. It is not inhomogeneous, it's partially inhomogeneous station, it becomes oscillatory. And that's the formation of the pink one that you are seeing in the last in H. The pink one is the limit cycle. That's the PD limit cycle.
Yeah.
And that's forming. But you can see when you go up in time, you have oscillations in time.
Is that what D is showing? Is this
Exactly.
Yes. Yeah.
Exactly. So it's going up in time as you go up, but there is still the pattern still dominates.
Yeah.
It's still, you can see the high concentrations in the middle,
Yeah.
the lower concentrations on one side, but they are really oscillations. So if you can see in F reflects exactly these oscillations. The L2 norm, once the limit cycle starts to fall. I'm not so sure this has been observed. That's why we are really interested in this. How does diffusion, and here we can see from a numerical point of view, whether in the Floquet theory, whether it can tell us this, and that's what we want to run with the Floquet theory, whether we can validate this observation. But there's quite a lot amazing dynamics that are happening. When we started with this, it was really amazing. You take one point, grid point, all of the grid points, they really behave very differently. All of them had different pathways of limit cycle. It's like you're sewing a carpet, for example. You have different threads. Individually, they form individual pathways.
Yeah.
When you put them together, they form a cycle. It was really amazing. And you don't see, I would like to see those type of results as well. Because they are really, this is what really started us, because we said if the system cannot behave to give you different solutions altogether per grid point. So you look at X0, X2, X3 as grid points. I think this was in 1D.
Yeah.
All this in 1D. So you could plot from 0 to 1, grid it, plot each X point and follow them. They give you completely very similarly, different results, but actually they are forming that limit cycle. And so if we can do the Floquet theory, maybe it might help us to understand again when diffusion drives the system unstable. But already we can see quite a lot of rich information in D. And again, we didn't understand this. So if you plot it, it really tells you exactly what is happening through the L2 norms and so on. But it needs to be described somehow. Yeah.
You should be able to compute the monodromy matrix. Not too difficultly for like when it's a spatially homogeneous system.
Okay.
When it's inhomogeneous.
Yeah.
Pain.
Yeah, because you have to compute it for every wave number. You have every D. Okay.
Yeah.
What information can you extrapolate? I would look at the spectrum, maybe. That's the only thing I can think of.
Yeah. I guess if computing the monodromy matrix for a specific K does not take that time to run,
okay,
or take that long to run, then you can do it for all the wave numbers and get like a dispersion relation curve.
Okay.
Values, which I don't think would be that difficult. There are quite a few different methods for computing a monodromy matrix. Unfortunately, time integration is not very good.
Okay.
Direct time integration is not very good. Right? Because all the fundamental matrices is, or what is it? Right, it's your solution components along.
Yes, yeah.
Although it might be fine in 2 components. That's what I tried once for my standing waves.
Okay.
It was terrible. Took forever to run, and that's wholly inaccurate.
Okay.
Which is why I gave up and started using autos.
But I think
Auto can do it too.
Okay.
Yeah. It's good that you two can talk about this. I think it would be nice to have some understanding of how.
Yeah, I have some papers on computing the monodromy matrix somewhere.
Okay, yeah. If you can get them, then yeah. Any other questions?
There's none on the shoot. It's with that. Thanks.
Okay.
I have a question.
Okay. Yeah.
Okay, I have a question. I'm doing something on. I have a question for my supervisor. I'm doing something on tumor progression, the tumor growth, and the dynamics. So, since your model captures the reaction diffusion-driven contractility, my question is this: can this model help predict transition between the aggressive and the tumor that are dormant because they involve the bistability and something?
I'm not quite sure I understand the question.
Because, yeah, I think one is to look at the model. It's a bistable system.
Mm-hmm.
Okay. And yeah.
Yes.
Do you have similar dynamics to what we observed in terms of the uniform steady states existing in the present or limit cycle as well?
Mm.
For the ODE. So do you have results for the ODE?
No.
Yeah, I would do the ODE system first before you can do the PDE. I think you can talk to Victor. Yeah, you can do the ODE first and try to exhaust the ODE and try to understand what. In fact, you can apply the locate theory also to the ODE system.
Yeah.
So it will be much easier before you add space.
Yeah.
Just look at the ODE itself, and then depending on that, I mean, it depends on the structure of your system. If it exhibits similar dynamics, I think then it makes sense to do this. But if you have just a unique limit cycle without other steady states around, it might not work. I'm not so sure. I don't think it will be the same.
Okay, thank you.
Yeah.
Hey, Vincent has a question. I can see him.
No, I don't have a question, but I was just scared by the 4 weeks simulations. Which kind of laptop?
But you were scared by the 4-week simulation.
Oh, that's okay.
Yeah. That happens.
So that's
Yeah.
He's using his MacBook.
Yeah.
Yeah, because I tried my laptop at home. Then the whole day it could not even move one iteration, and then I had to stop it.
Yeah.
Yeah.
Yeah. Okay.
Okay, so if there's no more questions, I'm gonna end the Zoom
Yeah.
call.
Yeah. Thanks.
Okay. Thank you.
Okay. Right. Oh. Yeah.